So the next question, which states the crystal structure of a peptide has ordered structural repeat of amino acids. Okay, with a distance of 6.5 angstrom between the alternate C alpha atoms. So which one of the following pair of dihedral angles accurately represent the peptide structure? Okay, so as I have already discussed about the peptide bond and all those angles, okay, so the peptide bond I have already drawn. This is the peptide bond. Okay, so this is the peptide bond, which is omega angle. Okay, and there where the rotation is not very permitted, and here these two angles between C alpha and C, the psi angle, and the N and C alpha, which is the phi angle. Okay, here the rotations are visible, but because of the bulkiness of the different R groups, all the angles are not allowed. So there are restricted angles for phi and psi are allowed. Okay, so before going to the phi and psi and permitted angles or the most allowed angles, so let's talk about the secondary structure of the proteins. So we have seen the primary structure where the different amino acid residues form a peptide bond and gets associated with each other and forms a long polypeptide chain. So when the long polypeptide chain is formed, they try to make some intra or interchain bonding and try to become a compact structure. Okay, so in those compact structure, the next level after the primary structure comes is the secondary structure. Okay, so the secondary structure of the protein, talking about the secondary structure of the protein, let's begin with the structure of the alpha helix. So the most common type of secondary structure of the protein is alpha helix. So that in alpha helix, a hydrogen bond is being formed between the NH group to the CO group of the other amino acids. Okay, so these bondings, these hydrogen bondings are there. So there are different hydrogen bondings you can see. Okay, and the alkyl group of the alpha helix chain are not involved in the hydrogen bond formation but maintain the alpha helix structure. Okay, and this entire thing after the intrahydrogen bonds forming takes a form of a helix like structure. That is why it is called as alpha helix, and these are the hydrogen bondings present. Okay, so each helix involves 3.6 amino acid residues and total 13 atoms that takes part in one whole turn. So that is why the alpha helix also called as 3.613 helix. Okay, and in alpha helix, the most important point to note here is in alpha helix, the distance between two adjacent amino acid is 1.5 angstrom. Okay, so this is the basics of alpha helix. A similar another secondary structure which is found, it is not in form in the not in the form of helix, that is in the form of sheets, plates. So these are called as beta pleated sheets of protein, and is a type of secondary structure in the protein. It consists of various beta strands linked by hydrogen bondings between adjacent strands. And so here the beta sheets could be of two types. So here, if both the beta strands, so individually they are called as beta strands, and all together they form a beta sheet. Okay, so if two beta strands are in the same direction, then they are called as parallel beta sheets. And if they are in opposite direction and joined by the hydrogen bonding between each other, that is called the anti-parallel beta sheet. So in the beta sheet, we will find the inter-hydrogen bonding, while in the alpha helix, we find intra-hydrogen bonding. Okay. So there in the structure of alpha helix, the distance between the two adjacent amino acid was shorter because of the helix formation. So there the distance was 1.5 angstrom, but here in the beta sheet, the distance between the adjacent amino acid is 3.5 angstrom. Okay. So yeah. So this is the, next comes the Ramachandran plot. Okay. So as we have talked about the dihedral angles and their rotation around the around the single bonds, and the rotation of atoms around the single bonds, that depends upon the R group and side chain. So here in this structure you can see the C alpha and C, this bond present is psi, C alpha and N, the bond present is phi, and here is the peptide bond present. Okay, this is the peptide bond, and here is the dihedral angles for the rotation of the protein structure. So all the rotations are not permitted. So according to the Ramachandran plot, there are only few permitted uh, angles of the phi and psi. So this plot is divided in four different quadrants. So here one x-axis where we can see phi, there the values are from -180 to +180. Another y-axis, the values of psi are given from -180 to +180. So the total graph is divided in four different quadrants. So for, if I'll talk about the individual amino acids, so as of presence of the small or simple R group in the glycine. Glycine occupies most of the space in the Ramachandran plot, while the proline residue, because of this cyclic nature and involvement of amino carbon and C alpha carbon, very less restriction angles are provided, and very there are made restriction to the proline structure in the movement around these C alpha C and C alpha N bonds, and that's why the proline has the least space allotted in the Ramachandran plot. So talking about the overall amino acid structure, so these are the place allotted. So here the red area show the highly allowed phi and psi bond angles in the Ramachandran plot, while the yellow area is allowed angles and the white ones are the not allowed. Okay, so here we can see beta sheet is present in this particular comes in this particular quadrant according to the psi and phi angles. Right-handed alpha helix in this particular corner, left-handed alpha helix in this particular quadrant. So if we'll see our options, or if we'll see our questions again, so there it states the repeat of amino acid with a distance of 6.5 angstrom between the alternate C alpha atoms. Okay, so we know that in the distance between the adjacent C alpha atoms in alpha helix is 1.5. In beta sheet it is 3.5 angstrom. Okay, so it is talking about the distance between alternate C alpha atoms. So the distance between these two would be in range of 3 if the structure would be alpha helix. But it is given 6.5 angstrom. That qualifies it as a beta sheet because the distance for beta sheet between two alternate carbon, it will be approximately 7 angstrom, which is close to 6.5 angstrom. So with this particular point, now we are clear that we have beta sheet in our structure. So for the beta sheet, it has been asked the dihedral angles accurately represent the peptide structure. So the peptide structure is beta sheet. So now we have to find out the angles. Okay, so in the Ramachandra plot, this is the value of phi, value of psi, plus 180, plus 180, this is minus 180. So here comes the beta sheet. That is also in very this angle. Here the right-handed alpha helix, here the left-handed alpha helix. Okay, so the first option is phi and psi both are in negative. Okay, so phi minus 60, that means near here, and psi also minus 60, that means here in this quadrant, which is not at all allowed for any structure. Okay, so next comes another one, which is minus 120 and minus 50. Again, for the psi it is here somehow, and for phi it is here. So that also for the alpha helix, not for the beta sheet. So this option is also wrong. Third one is minus 60 and plus 120. So minus 60 for phi, that means somewhere here, and plus minus 120, which is somewhere here. So here and here in this area, there also nothing, no movement would be allowed, no. So this option is also wrong. The fourth option is minus 120 for phi and minus 120 for psi. So that will come under this area, which is covered by the beta sheet structure. So for this particular polypeptide, this particular dihedral angles could be possible. Okay, so that's how these questions can be solved, knowing the exact Ramachandra plot where which secondary structure is allotted and the values or facts related to the secondary structure of the amino acid. And this is my NPTEL reference. Thank you.